Hi, ChangHui.
Hi, Aaron. How are you? Can you hear
Mm,
me? Can you hear me okay?
I can hear you. Yep. Can you hear me?
Yeah.
Great. Thanks for coming.
Yeah. Thank you for having me.
Of course. Do you want to test your slides out?
Mm, my screen okay.
Yep. Looks good.
Progressing fine.
Hmm.
Okay, perfect.
Yeah, looks good. How was the rest of your summer?
Good. So I flew from LA to South Korea for 2 weeks and then brought my kids back to
Okay.
US. Good.
Uh-huh. How was that?
That was that was good. It was very hot and humid, but yeah, it was good to see kids very happy living there in away from parents for 2 months. So that was a good sort of reunion. So
Wow. Been my dream for like 14 years to like send my kids away for like 1 or 2 months.
yeah, I tried this for the first time and it was amazing. I think both ways.
I would just take it for like one weekend even.
Yeah.
Hey, Lulu.
Hi, Aaron.
Good to see you.
I'm good. Okay, great. I tried to fix my internet connection. Hopefully, it's gonna be better.
Seems okay.
And
Yeah.
if I stop freezing, because I have some movies, but if I stop freezing, please let me know so I can kind of like probably shut off my video screen. But other
Okay.
than that, yeah. You're
Okay.
in China right now?
Yeah, in Shanghai.
Oh,
Where's my dog? Oh,
fun.
there's my dog.
Hey, what's your dog's name again?
Archie. Archie. Archie, say hi. Say, come on. Let's go over there. Yeah, Archie, come on. Yeah.
Oh, so cute.
Archie, say hi. Oh,
Hi.
oh, she wants to go. She wants to go. Okay. Yeah, that's Archie. Oh, she went there to her little bed.
Cute. Oh,
Yeah, Archie. Okay. Do you want to test out your
yeah.
slides?
Yes.
Aaron,
Hi, it's long.
hi, Lulu, ChangHui. Nice to see you.
Nice to see you.
Can you see my slide?
It's do the
Can
yes,
you
but
see my slides?
do the slideshow view.
Okay. Slideshow
Like
view. Yeah,
or
I am. I'm not.
I don't see it. Or
Lulu,
maybe
I think you're freezing again.
I okay. Okay. Yeah. Let's do this again. So I tested earlier, and
you might have to like swap displays or something.
yeah. How about okay. Hold on.
Mm, It says started screen
Yeah,
sharing.
so when I start, it freezes a bit. So if it's okay, I can leave my. Yeah, I
It's
start
working.
already.
Yeah, sure.
Can you-
Yeah.
Yeah.
oh, probably there's a delay.
Can you go to the
Yeah.
next?
Is it full screen?
Yeah.
Yeah.
It's good. But try like a couple slides just to see. Can you click through to different slides?
Yeah, I am.
Oh, maybe there's some sort of a delay. Oh, for us.
Okay.
Now we see it.
Yeah, I think there's a delay.
So maybe it's a
actually
network
moved
issue.
from my office because my office somehow it's even worse. But still, I think it's probably too much movies. But I just tested again and it was fine. So
Do you have like
maybe I'll
some
take
other
the time
network?
when I change my. Right now, no.
So you're in your office or you're in your home?
Is this you can see I'm changing my slide.
No.
Right now I'm at home.
No,
Yeah,
maybe
I then
network
moved
issue, I
to
guess.
home
Oh,
just so then to get a better reception.
Um,
Yeah. Wait, so you can see I'm switching my slides right now, right?
Right now we only see the title slide.
Okay.
Now we see the slide number 2.
Okay. Yeah.
Now we still see slide number 2.
Okay. And I just clicked to 3.
I don't see it yet.
Okay. Yeah, I'll probably just try to
Now we
maybe the
see
network is a little slow. Yeah, I guess all the problem is.
Can you switch to your phone, your data from your phone instead? Is that faster?
maybe probably accept that. Not that straightforward.
Okay.
Yeah. So I just lost.
This is
my
like
phone
you're on campus.
recently.
No,
I mean,
this is you're on campus.
I'm on campus. That's why. Yeah, I'm still on campus. So I think that's probably the problem. I was even calling the IT people trying to fix this issue. Yeah, I'll go back to first slide. At least I think
We can hear you. And
at least I'm not freezing, right?
now the audio is pretty poor.
Am I freezing? I'm not freezing. It's just the slides are slow. Oh,
Yeah.
shoot. Okay. Yeah, I'm so sorry.
Is it the Wi-Fi or the network? Or you?
No, right. It's actually I think it's just the network that's having issue. But like I said, I just fixed, but I guess it's still.
I think it may be helpful if you turn off the camera.
Yeah, let me try that. Hi, Pascal. Okay, let me stop my video. Maybe that helps. Let me see. Now I go to slide 2. I guess there's just a delay.
We don't see slide 2 yet.
Yeah, I'm so sorry about this. No slide 2.
Now we see slide 2.
Yeah, but this delay is so bad. I'm so sorry. Yeah, slide 3.
This is okay. It's good training for giving talks.
It's not moving to slide 3.
Not yet.
Yeah, it's so too.
Yeah, because actually our lab building recently have issues. So we have been having bad internet in my building. I always have to, for more important stuff, I have to come home. But
Do you
I
have
don't know. It's
do you have
just,
data
I
on
think
your
it's
phone?
more of a UCN work. You are seeing my slides.
Yeah, slide
Do it
3.
off my phone. Yeah, I just.
Do you have 5G on your phone?
Yeah. Okay. Yeah. So it was like. Yeah.
Yeah, I think it's working. Just as a network, if you Lulu can use the hotspot on the cell, it should be fine.
Yeah. Okay. Let's see.
Yeah, just connect to your cell phone data instead of
Let's see if this person-
the
Right.
campus network.
Okay, I don't know what I'm doing. Sorry. Okay, let's see.
Should we start with
Sure.
ChangHui instead, while you sort out the problems, Lulu, or do you think it'll be okay?
Sure, sure. If
Sure.
that's okay with ChangHui, sorry about that.
No
Okay.
problem.
Yeah.
Okay.
Great.
Yeah. Okay. Yeah. So Lulu, just see if you can work on the network connections, and meanwhile, we'll get started. Welcome everyone. Another week of Neuro Zoom. Another to Sudhoff Lab former trainees. More coming. Thanks so much, and looking forward to their talks. Next week we have great talks from Bing-Sing Pan from Nanchang University and Seth Blackshaw. So tune in. Always exciting discussion and new insights. And please let Zalong and me know if you want to give talks about your research here. Okay. Zalong, can you introduce Dr. Pak?
Sure. It's my great pleasure to introduce Dr. ChangHui Pak. So she has her bachelor degree from UIUC and has a PhD in Emory University, working with Professor Colbert and Monberg. And in postdoc, she went to Stanford, working with Tom Sudhoff, and starting a very exciting field where I still remember the first couple of papers in the field to start use neurogenein to and among others to transform IPS to neurons. That's very exciting field. Later on, so ChangHui started her own lab in UMass and continued to use the human IPS derived neuron to study a series of important molecules, including neurexin and others. So today, ChangHui will bring us her latest dissecting the molecular and cellular mechanism of schizophrenia risk variants, neurexin-1, the leading use the human-induced polypotent stem cells. Welcome, ChangHui.
Thank you so much, Zalong, and also Aaron for the invitation. I will start sharing my screen. Let me see if this works. Okay, great.
Yeah, they're putting
Awesome.
the
Yeah,
sure.
can you see my screen?
Take it away.
Okay, perfect. Okay, thank you so much for your kind introduction and also your invitation. So like Zalong mentioned, I'm going to be talking to you about dissecting the molecular and cellular mechanisms of schizophrenia risk variant, neurexin-1 deletions using human-induced pluripotent stem cell models. So let me just dive right into it. Due to the sort of the advances in the GWAS as well as exome sequencing studies from individuals with schizophrenia and controls, we now have a sort of a better and a clearer picture of the genetic contribution of schizophrenia. Here you can see a figure from Singh 2022 paper where it outlines these presence of these common variants, which are single nucleotide polymorphisms, which act in aggregate to increase risk for schizophrenia. Also to the left here, where we have these copy number variations, which are usually present as copy number deletions or duplications, losses or gains in these chromosomal regions, which are less frequent in the population, but as these copy number variations, once one has them, dramatically increases the likelihood of developing this disease. And on top of that, we have now put also these protein truncating variant mutations, which are missense damaging mutations in individual genes that also increase risk for the disorders. And the top 10 genes are actually highlighted here on the left in these red dots. And this is really an exciting time for us because of this integration of both the genomics as well as these technology to convert somatic cells, either blood cells or skin biopsy, skin fibroblasts of living patients with these disorders into human-induced pluripotent stem cells. And what this allows us to do is to capture and study these genetic variations from living patients in vitro in these systems. At the same time, we can use gene editing tools to engineer various mutations that are so significantly associated with these disorders. You can engineer SNPs, common variants, as well as copy number variants and protein truncating variants to study their functional contributions of these risk variants in isolation in an isogenic background in a controlled manner. So from both of these studies, one can then ask the following questions. What are the underlying biological mechanisms of these disease variants, and how can they contribute to these diseases? And so sort of the focus of my talk today will span this particular CNV, 2p16.3, which is a copy number deletions that is associated with schizophrenia. And interestingly and conveniently, this chromosomal region, this deletion spans one single gene, and that is the presynaptic neurexin-1 cell adhesion molecule. Unlike these other CNVs, which actually encompasses dozens and dozens of genes, which actually sort of complicates the analysis because of its pleiotropic effects of multiple genes at play. This particular copy number deletion is really convenient for us because we can then assess the functional effects of a single gene in this and its association with disease. So neurexin, as many of you already know, are central synaptic organizers to specify and maintain synaptic efficacy. In our human genome, there are 3 neurexins, 1 through 3. And we know also that alternative splicing of these molecules allows these diverse partnerships across synapses, allowing these molecules to perform very diverse functions across brain regions and synapses. And for reasons that we don't quite well understand, neurexin-1 deletions are disproportionately represented in neuropsychiatric diseases. And that's not just schizophrenia, but also in autism, intellectual disabilities, Tourette syndrome, and Pitt-Hopkins-like syndrome. And also, I think it's important to note that not only neurexin-1 deletions are impacted in disease, but also these various genes that actually participate in the neurexin complex formation and signaling are also found mutated in these diseases, suggesting that the neurexin complex formation and signaling can be a shared mechanism across these various disorders. And we know that synaptic dysfunction is a key underlying mechanism for various neuropsychiatric diseases. So today I'm going to talk to you about two in two parts. One is our heroic effort across multiple sites and involving multiple PIs, in which we've established and analyzed the functional contribution of neurexin-1 copy number variations, these deletions, using schizophrenia patient-derived iPSCs, as well as the neurexin-1 conditional knockout iPSC model. And this study was published last year. Then I'm going to follow up and in the following slides, talk about our more recent work that's actually unpublished, work from my lab, a graduate student, Rebecca, and a graduate student from Bruce Arono's lab, Kang-Jin, which was a joint effort in using single-cell transcriptomics to understand the developmental and cell-type specific contributions of neurexin-1 CNVs in developing brain organoid models. So let me start with the first part. We've used the molecular genetics of schizophrenia European ancestry cohort blood samples that was collected many years ago. And we've established iPSC lines from individuals with schizophrenia who are natural deletion and duplication carriers for these different CNVs. So we've established 5 individual iPSC and multiple subclones from these individuals who carry neurexin-1 deletions, as well as individuals with 22q deletions and individuals with 16p 11.2 duplications. Moreover, we've established iPSCs from control donors who are healthy and unaffected, and these were randomly paired with these other patient-derived iPSCs. Upon reprogramming these blood cells into iPSCs and establishing these lines, this actually reprogramming was done in a single site, the RUCDR, which is now called Infinity Biologics. These cryovials of cells were then dispersed across multiple sites. And the first location, obviously, was at Stanford, where we've, in a single lab, academic lab setting, grew these cell lines, expanded them, banked them, and also used these iPSCs to differentiate into cortical-induced neuronal cells. And as Zhilong mentioned, these are NGN2 cells, for those of you who are not familiar. These generate, this method uses a single transcription factor to generate a relatively homogeneous population of cortical glutamatergic neurons. And using these cells, we've then analyzed their morphology, the physiology, and also their gene expression. And at the same time, these iPSC vials also went to Fujifilm Cellular Dynamics, Inc., a company that is an expert in making differentiated cells. We partnered with this company, and we've developed a method to really scale up IN cell production such that they were able to produce induced neurons at industry scale and send these vials of neurons to us to multiple sites, including Rutgers in Ziping Peng's lab, where his group performed independent parallel analysis and looked at morphology and electrophysiology. At the same time, using one of these control donor cell line iPSC, we've engineered a neurexin conditional knockout heterozygous mutation. Here's a schematic of the targeting strategy, where we place LOXP sites surrounding exon 19, which is the first shared exon between the long isoform alpha and the beta. And using the appropriate recombinases in cell culture, we can then conditionally generate a wild type control or a heterozygous knockout. So the point of this sort of study design, and why I'm telling you sort of the overall structure of the study design, is that we really wanted to address the scalability, reproducibility, as well as rigor. And that's something that we really wanted to achieve from the study, as well as understand what is the functional consequence of these neurexin deletions in schizophrenia patient-derived neurons. So what have we learned from these iPSC models? So these schizophrenia neurexin-deleted neurons, when we looked at their morphology, as shown here in these GFP-filled single neuron pictures, we did not observe any change in their dendritic outgrowth or in their neurite outgrowth morphology. And the quantifications are shown in the middle, where when we assayed all kinds of parameters, as shown here, we did not detect any change. Nor did we detect changes in the density of synapsin positive puncta or their puncta size, suggesting that synapse formation in these cells is normal and their neurite outgrowth. This result was replicated across sites, again at Rutgers, using different cell source as well as different analysis software to analyze these morphology. However, when we patched onto these neurons and performed electrophysiology experiments, we started to see, we saw a very specific synaptic impairment. And these are shown in this figure, where in these neurexin schizophrenia deleted neurons, we saw a specific decrease in the miniature EPSC frequency, but not in the amplitude, suggesting that perhaps neurotransmitter release probability could be altered. And again, the same phenotype was observed across sites at Rutgers as well, using independent measures. And when we measured action potential evoked responses, evoked EPSC amplitudes were consistently decreased in multiple disease lines, suggesting that their synaptic strength is decreased. And when we performed pair pulse ratio experiments, where we delivered two closely spaced action potentials and calculated their pair pulse ratio, which is an indirect measure of neurotransmitter release probability, we saw a consistent increase in the PPR, suggesting a decrease in neurotransmitter release probability. And this phenotype, again, was replicated at Rutgers as well. So what about neurexin deletion in an isogenic background? What I didn't tell you is that we've actually validated this neurexin-1 conditional knockout strategy in a human ES cell background back in 2015, and we actually saw a very similar synaptic impairment. And what we wanted to do was basically engineer the same exact mutation, now not in an ES cell background, but in a control donor iPSC background, to figure out whether these neurexin CNV in an isogenic background elicits the same phenotype. And what we saw was that these engineered conditional knockout mutation in these controlled genetic background actually recapitulates the synaptic transmission deficits that we saw previously in the patient-derived cells, and also replicated the results from the 2015 work. So combining this result in addition with the previous results that I've shown you, then we can definitively conclude that neurexin-1 deletions, regardless of the genetic background, elicits a specific synaptic impairment in human cells. Finally, we performed the same exact experiment, now using a mouse line, which actually harbors the same exact conditional neurexin-1 alpha beta mutation. And we isolated mouse ES cells from these mice and performed the same type of differentiation experiment so that we can produce the exact same ESIN cells in the mouse, from the mouse, and perform the same functional experiment. And what we saw was that there was no phenotype that could be observed in these mouse ESIN cells, suggesting that this synaptic impairment is specific to human cells. So that kind of sums up the first part of my talk, where we've now, I think we can really definitively say, at least in these cell types that we are differentiating or making in the dish, that there is a specific synaptic impairment associated with these neurexin-1 deletions that are present in both the isogenic control background as well as the patient-derived background. So there were a couple of questions that were still lingering in my mind, which were the following. Because these induced neurons actually skip all the necessary neurodevelopmental steps to get to these neuronal states, and because we just fast forward the differentiation using transcription factor overexpression, we have this gap in knowledge from these ES or iPS cells and how they progress through neurodevelopment. So we asked, what are the effects of neurexin deletions in diverse cell types during early cortical formation and neuronal maturation, and ultimately to forming these synapses? And second, are there developmental stages and or cell types that could be most vulnerable to these neurexin-1 deletions? And third, are these effects genetic background specific? And to answer these questions, I think we have in the field now a great developmental model. These are the brain region specific organoids, where we can actually capture these sort of progression of neurodevelopment in 3D in the dish. So utilizing such protocol, using the same set of lines that we've analyzed their synapses in the previous part of the talk, we perform single cell RNA sequencing using 10x genomics. And in sort of trying to reveal whether and if there are any developmental timing and cell type specific perturbations that could be induced by these deletions. And so we really wanted to create a single cell transcriptomic map across different mutation status and across a different developmental time. So what does the data look like? So this is an integrated UMass. Each dot is a single cell projected onto a 2D space. And these at the end of the day, we have recovered about 107,000 cells that have high quality data passing QC. And we've collected brain organoids from 3 weeks, 50 days, and to about 3.5 months across these developmental times, and also across genotypes and organoid types. So donor here means the schizophrenia patient-derived organoids, and engineered means these are isogenically engineered neurexin-1 conditional knockouts, organoids. And what you can see is that we have produced all the predicted cell types that one would expect to produce in these brain organoids from these developing neuroprecursor cells that are both dividing and non-dividing, as well as these intermediate cells then differentiate into specific neuronal subtypes, including the cortical glutamatergic cells and GABAergic neurons. In addition, we have some presence of glial cells and astroglia as well in the prep. So we are quite happy with the quality of the data, and we then can use these cells to analyze and ask specific questions. So because of limited time, I'm going to just give you the punchlines, and I want to sort of frame it in terms of the differences as well as similarities that we saw from these analyses. So first, let me focus on the differences. So we observed in this very busy slide that these are the isogenic conditional knockout organoids on the left, and these are the schizophrenia neurexin deletion organoids to the right. And what you can already observe is that the number of cell types that actually have differential gene expression patterns in the isogenic organoids are quite modest and in number, as well as the sheer number of DGs that are present in these cell types, as well as the overlaps of these DGs across cell types. And I'm omitting these 3-week-old data because pretty much it was very quiet there. Neurexin deletions, it doesn't seem to perturb early progenitor cells as much. And this is something that was really surprising to see in the patient-derived organoids, because as early as 3 weeks in culture, you could already see that there's a lot of massive gene expression changes going on from 3 weeks to 50 days to about 3.5 months. And these changes in gene expression are definitely greater in magnitude, and also the overlaps between these cell types are also very much shared in great number in the donor-derived organoids. And I'm not showing you the data here, but through proportion data analysis, we've also have seen that actually these neuroprecursor cell changes are actually very specific to this genetic background, suggesting that neurexin deletions do not, in isolation, do not cause changes in neuroprecursor cell changes, and that it is driven by these schizophrenia genetic background effects. Also, what you can do is take these data set and perform what's called a pseudo-time trajectory analysis. And this is all it is putting these cells onto pseudo-time and actually tracking these changes as a function of a progress the cell has made through differentiation. And what you can also observe, similar to what I showed you in the previous slide, is that in the isogenic brain organoids, these control and conditional knockouts conditions are very similar across their developmental trajectory across pseudotime. And only in the more maturing and more maturing brain organoid age, you start to see some deviation of their developmental trajectory. However, sort of mirroring these gene expression deficits or gene expression changes in the schizophrenia deletion background, at around day 50, you start to see these changes and shifts in their developmental trajectory, which is quite obvious by the time you reach about 3.5 months. So we observed that the timing of changes in these cells' developmental trajectories were different. What were the similarities? What we observed is that the maturing glutamatergic and GABAergic neurons seems to be the key cell types that seems to be most vulnerable to these mutations across genetic backgrounds. And that when you look at sort of these enrichment of these differentially expressed genes, what you can see is that these genes actually sort of fall into specific biological pathways. And what we were sort of predicting to see were obviously changes in genes that are associated with synaptic signaling, which is gratifying to see. However, we interestingly also observed that the RNA splicing machinery, as well as factors that that act in the ubiquitin proteasome pathways, were also consistently altered in both genetic backgrounds. And this is actually quite interesting, and I think exciting for us, because all three of these pathways have been implicated in schizophrenia biology. And changes in these gene expression actually then also translate to long-lasting changes in neuronal network connectivity. As one can see here in these 5-month organoids, as they mature, we perform calcium imaging experiments, live calcium imaging experiments, and you can already see that the neuronal firing, both in the spike data as well as the synchronicity of these spikes as measured by these calcium transients, were actually changed and were decreased in the schizophrenia neurexin deletion background. And also in the engineered conditional knockout background, we actually see an increase in the neuronal spike frequency. Again, suggesting that, yes, indeed, towards the end, we do see changes in long-lasting changes in neuronal connectivity. So with that, I'd like to acknowledge for the first part of the talk, multiple PIs and members who've contributed to this work, including Thomas Sudoff, my postdoc mentor, who's been my mentor for 7 years, as well as the funding sources at NIMH, as well as the POC lab for the second part of the talk, members past and current, and collaborators, as well as funding sources. So with that, thank you so much for your attention, and I'd be happy to take any questions.
Thanks so much, ChangHui, for a beautiful talk. So open for question. So ChangHui, do you want to ask a question yourself? Put a question on the chat board.
Yes, excellent work, amazing. My question is there data or your own data in the human postmortem brains to validate your finding from these epilepsy-derived brain organoids?
Yes, no, that's very a great point. So we do not have postmortem data from neurexin-1 deletion carriers. However, we actually did some comparative analysis of postmortem embryonic brain tissue, which actually was published by the Arnold Krickstein's lab by Baduri et al. And we actually did sort of side-by-side comparison and using the control donor-derived organoids, because we wanted to see, where Neurexin-1 mRNA is most expressed across embryonic brain development. And what we can also, and we did some comparative analysis with that, but and it seems that definitely there are similarities, but also there are differences. And just as a general sort of, I guess, a word of caution is that these brain organoids, although they're excellent, I think, cellular in vitro systems, but also they definitely are sort of their own beast, meaning that although you can observe similarities with actual postmortem tissue, definitely there are differences.
I got a question for the difference compared to the isogenic versus the schizophrenia-derived neurons. So would you able to, I think there's light and neural projections. So would you able to observe any anatomical evidence supporting that Neurexin-1 deletion has some impact to the neural projection process? Because as I know, for the schizophrenia, it normally has very mixed background, right? So Neurexin-1 may be the common shared variant, but more likely each patient might have some individual variants by themselves. So I'm curious about whether the neural projection is a common defect could be discovered by your analysis.
Right. So actually, let me maybe share this. Sorry, I quit sharing too early, I think. So this is sort of in the supplementary figure. So this is a cell proportion analysis that we performed. This actually, to the left are your Neurexin conditional knockouts, and where you can actually quantify its differences in cell proportions of these organoids. Again, this is actually sort of underpowered, and I think we need more samples to this, but you can actually start to observe some differences in their cell proportions. And this is, again, sort of very different than the schizophrenia Neurexin deletion background.
Right.
There are definitely changes in the intermediate precursor cells and their sort of distribution of these inhibitory and excitatory cells in various different cell types. But we know that we also, again, have to be careful with these kind of data because there is an inherent variability across, batches and lines, even in line genetic backgrounds when we are looking at these proportion data. So I think what we need to do is perhaps increase the sample size and perhaps use multiple Neurexin conditional knockout organoids, and to really definitively say sort of what kind of sort of developmental trajectory defects we're seeing. And I hope that answers your question.
Sure. Okay, great. Yes, and any more questions? Okay, if no more questions, thanks ChangHui for her beautiful talk. Thank you. Let's start from Lulu's talk. Aaron?
Great. Thanks so much, ChangHui. And it's a great pleasure to introduce Dr. Lulu Chen, our next speaker. Lulu is an assistant professor in the Department of Anatomy and Neurobiology at University of California, Irvine. I first met her at an In-N-Out Burger restaurant in Ventura, and when I saw that she could order off the secret menu, I knew that we could be friends. And it's been great getting to know her. She's from Taiwan, and then she did her undergraduate degree at Cal State University, Long Beach. She then did a PhD at UC Irvine, with doctors Christine Gall and Gary Lynch, and here she studied activation of signaling pathways during learning. In an interesting paper during her PhD in the Journal of Neuroscience, she discovered that during novel object learning tasks, BDNF signaling and TrkB receptor activation is differentially affected in different regions of the hippocampus. She then did her postdoc at Stanford with Dr. Tom Sudhoff, and here she worked on neurexins. These are key molecules that we've been learning about this month in the formation of synapses. There are 3 of these in mice, and it was unclear if they each play the same role at all synapses or they have different functions. And lots of papers we say are an amazing paper, but this is really an amazing paper. It's published in Neuron. Lulu solved this problem really the only way you could do it. She generated with her colleagues triple conditional knockouts for all 3 genes, and then deleted these in neurons and analyzed phenotypes at 3 different synapses. And I guess contrary to expectation, she found that each synapse had a dramatic phenotype, but they were different based on context and neuron type. So this led to the conclusion that they don't perform standard functions, neurexins, but rather distinct functions based on distinct synapses. She then was recruited back to the same department she did her PhD in at UC Irvine. So this is a good lesson for graduate students. If you do well in grad school, you might get recruited back to that department. And I guess you can use the same email address as well. It's another advantage. And since she's been there, her lab is interested in identifying the key molecules and signaling pathways involved in synapse formation and learning and memory and disease as well. So looking forward to hearing the latest, Lulu.
Hi, thank you. I hope Aaron will still be my friend after having so much technical issues. And right now, I'll try to project this from my phone, and hopefully we don't have any issue. And please bear with me. And yes, hi, it's absolutely honored to be here with such a great audience. I'm Lulu Chen. I started my lab in anatomy and neurobiology at UC Irvine October 2018. So 4 days. I will reach a 4-year anniversary. So I'm very excited to share with you the most recent work from my lab. So kind of a very brief introduction about neurexins. Thank you, ChangHui, for being flexible, and she also introduced neurexins. Okay, yeah, I think I froze a bit, and it's okay. Neurexin background, we kind of all know neurexins are very important, regulating synapses, regulating synaptic function, how they actually assemble. And there's 3 genes. Each gene has the alpha and beta isoform. Neurexin 1 has the gamma. So these proteins and genes are highly implicated in human psychiatric disorders. So as Aaron was mentioning, that when I started my postdoc, we did this conditional knockout mice of neurexin 1, 2, 3, alpha and beta. So this allows postnatal survival, and also we can really look at with the spatial and also temporal control. So long story short, what we actually find is that even though neurexin 1, 2, and 3, they have canonical very conserved domain structure. However, neurexins, they actually have very different function that depending on what synaptic context that they're in. So, so this kind of leads to neurexin 2, even though neurexin 2, it's, neurexin has been discovered by Tom Sudhoff like in the early 90s, but neurexin 2 still remains the least studied neurexin. In the last 5 years, there's increasing studies show that neurexin 2 mutations, growing evidence showing that they are mutation in patients with autism spectrum. Neurexin 2 also expressed in different regions and different cell type. However, until now, we really don't have a general idea what neurexin 2 is doing, or whether, if they have any cell type specific function. So during my postdoc, and also with the Thomas Sudhoff lab, many lab members in the lab, that we characterized neurexin 2. And what we found is that using this conditional knockout mice of neurexin-2, we found that neurexin-2 not only is different from neurexin 1 and 3, it is quite opposite from neurexin 1 and 3, meaning that in the hippocampus, we discover neurexin-2 inhibits excitatory synaptic connections and functions. So when we delete neurexin-2, we actually have increase of excitatory synaptic function, frequency, and also synapse density. Now, when I started my lab, my first question is, what if we delete neurexin-2 in the excitatory neurons? Other words, meaning what happens if we remove this inhibition in the excitatory synapses? So we did this with the neurexin-2 fox mice crossed with the EMX1 Cree. And this EMX1 Cree, they're actually expressed in the pyramidal excitatory neurons in the forebrain cortex and also in the hippocampus. So this allows me to actually ask a question that, region and cell type specific knockoff neurexin-2. And we first do RT-PCR to confirm that this neurexin-2 deletion is region specific and it does not alter the mRNA expression of neurexin 1 or 3. And we did not observe any kind of general body weight or brain weight change in these neurexin-2 specific knockout. And then we set out to look at the autism spectrum disorder core symptom of these disorder usually, and so that is social interaction and also repetitive behavior. So what we see is that in our knockout, they prefer to interact with the peer mouse more than object, just like the wild type with the object. Then with the wild type, the wild type spend the time with the object. So we see this that they have a preference to interact with the object more. Moreover, when we look at necessary shedding, we saw that they actually shed 2-fold more of the necessary than the wild type. However, we didn't see any marble-bearing differences. And probably you guys know, there is a debate whether we should actually consider marble-bearing tests as repetitive behavior or anxiety. And additionally, we look at those associated symptoms of autism spectrum disorder, and we find that in general, they're actually quite normal. We don't see any kind of, we don't see much of a change in the distance travel, or we don't see much differences compared to the wild type in elevated plasmates when we look at anxiety or light-dark box transitioning. Moreover, when we look at object recognition memory, we also see that they can learn simple tasks, very simple object recognition memory. Although we don't see these, and these are analyzed, however, I kind of still want to point out that we do see these knockout mice, to a greater extent than wild type, that they engage a lot of other behavior that are not task related. So for example, this is a marble bearing that these mice is actually doing a lot of digging and jumping instead of burying. Or this video now here, this mice is doing a lot of jumping on the corner, or this one, object recognition memory. This mice actually, it's climb on top of the familiar object instead of, exploring it, or this is how they explore. And also very interestingly is that when we look at the elevated plasmates, our mice, when it's in open arm, they actually jump off from the open arm. And this is actually very rare, less than 1% of mice, they do this behaviorally. So it just disappeared. So my point is that, as I said, these actually are removed from the quantitative analysis, but I still think this qualitative observation is quite interesting. So as we know that neurexin-2 encodes a adhesion molecule that has synapses, and synapses is actually very important for memory formation and how an animal learn and then store memory. So we set out to look at dendritic spines by doing Golgi staining, as we know, dendritic operization or density or maturity, these are also the hallmark for autism spectrum disorder. So we didn't really detect any big operization differences, and we did not see a change in the spine density. However, we see a trend in the mature spine, less mature spine in the consensual knockout, but significantly we have more immature spine. So, this shift between immature and mature spine could disrupt the synaptic function. So we also use wholesale patch clamp recordings from CA1 pyramidal neurons in the hippocampus, and we record this is spontaneous EPSC, excitatory postsynaptic current. And what we see is that we see a lot more frequent EPSCs, spontaneous EPSCs in the Neurexin 2 knockout, while the amplitude is intact. And also these events, they have smaller, significantly less decay tau, meaning that they go back to baseline a lot faster. So this kind of spontaneous events that, because we did not block on sodium channel, so you could have like 3 potential reasons that cause the increase of frequency, right? So it could be postsynaptically, but we look at the MND ratio, ratio, and we didn't see a change even then, but we don't really see a ratio change. So unlikely it's from postsynaptic. Presynaptic release, we also look at PPR. We don't see any kind of increase of a release probability from these knockout mice. And a very recent study from Sudolph lab, they also showed that these Neurexin triple knockout, they actually the EPSC also release GABA through regulation of GABA B receptor. And again, these Neurexin 2 knockout does not have show the same phenotype. I guess, again, this kind of shows that Neurexin 2 could be very different from Neurexin 1 and 3, or even when you look at it as a whole, Neurexin triple. And also, this kind of comes back to the conclusion that we think we probably have an overall increased network activity. There's other ways that we can test this to further investigate this, and we can talk about more. Okay, so if we have an overall increased network activity in vitro, this could indicate maybe there's an increased risk for epilepsy in vivo. Especially, we know that Neurexin 2 mutation has been repeatedly found in epilepsy as well. So we did a PTC injection. So PTC is the GABA A antagonist. So we inject this, and I will start the video. Hopefully it does not freeze too much. So these mice are genotype are blind, but as you can see that we basically do simultaneous injection on these knockout and wild type. And what we find is that it takes a lot less injection for the knockout to reach seizure. As small as as few as 6 injections can lead to a knockout seizure, or where it takes at least 13 for wild type. It takes a lot less time. And even though they're blinded, I guess you can tell which one is a knockout. If not, give it 2 more seconds. Yes. So the top is our Neurexin 2 knockout, and they have a higher susceptibility to drug-induced seizure. So we're curious about what about the spontaneous electrographic seizure, which is the hallmark of epilepsy. So to do this, we implant headsets that can allow for EEG and EMG recordings. And we do this for 24 hours a day. We record 2 to 4 weeks. And we do EEG, EMG, and also video recording. Okay, and we actually raised our analysis criteria to make it even more stringent, because as you know, patients can have a 3-second of seizure. But I think 3 seconds of seizure is actually, well, at least for me and a young lab, we're not as confident. So we make sure that all our seizure activity lasts more than 30 seconds. Okay, and so this is the EEG recording that we see, wild type, but for the neurexin 2 knockout is quite clear that we have a more than 2 times of amplitude, increased frequency, has a clear onset, and also a very clear, sharp termination of these seizure EEG activity. And as I told you, these are time locked, right? So I'm going to show you a video that is time locked with this particular trace. So that's this video, and as you can see, the mouse is going through these behavior stage raising scale. So now it's standing on top on the hind lip, and then it's falling, and it's gonna end up with a wild jumping. Okay, so yeah, so that's the neurexin 2 knockout. So we actually see this in 50% of our knockout mice. Well, with this very stringent criteria. And I also want to point that these are reoccurring seizures. So it's not just like happen once. More so, for example, this one animal has 10 reoccurring seizures, behavior seizures, spontaneous seizures that happens during the recording time frame. So I think all these data basically take us to a one sentence summary that is statutory neuron deletion of neurexin 2 in the forebrain results to in autism-like behavior abnormalities such as social interaction or preservative behavior patterns, and also spontaneous reoccurring seizures. And these are likely arising from the increase of hippocampal network. And also I want to point out that these EMX neurexin 2 knockout mice are, it's quite rare when you look at in the literature to look for epilepsy genetic model that these are non-fatal seizure and they are spontaneous. So it's not injury or drug-induced seizures, and they also are behavior scale seizures. So it's not just a song seizure that a lot of time that you observe. And of course, as I mentioned, neurexin 1, 2, or 3, they all have been implicated in epilepsy. Their mutation has found in patients that either develop seizures or were diagnosed with epilepsy. However, this is the first mouse that we observe spontaneous seizure. And I would say this probably signifies the importance when we're actually investigating, whether it's circuit or cell type specific function of these really important multi function molecule. So last, I'm going to thank my lab. Want to highlight she's my first graduate student, and she recently advanced to PhD candidacy. And my lab, and also Dr. Hunt from the neighbor lab, who gave us advice on these epilepsy recording or interpretation or criteria. And also the Thomas Sudoff lab, who, I work with all this exciting neurexin mouse models. And I think I just froze again, but yeah. And Dan, can I take your question? Sorry.
Thanks, Lulu. Great talk. And we're open for questions. Did you? Yeah. All right. Maybe they didn't intend to raise their hand.
So I do have a question. So I was not freezing too much this time.
Not too much.
Okay, a little bit. Okay, I'm sorry.
Yeah, okay. So, do you expect to see similar seizures with the other neurexin conditional knockouts with EMX-CRE, or haven't tried?
Yeah, so I'm absolutely interested in, especially want to understand like what is particularly neurexin 2 is doing, right? Because it's quite interesting, has this inhibition on exactly specific circuit, but I think I think yes. If we look at other neurexins, probably they do play a specific role in certain circuits, which I think hippocampus is a good place to go because that's usually where the seizure arise. Particularly, I think just neurexin 2 because I'm neurexin 1 or 3. I'm not so sure. I know neurexin 3 that, from Jason Alto's lab, they actually overexpresses in vitro one of the mutation. They see that it inhibits excitatory function when you overexpress neurexin 3. But I think it's probably, I could have something if I do manipulate neurexin 2 in a different, say, cell type or even circuit. It might still give me different answers or same.
Got it. Okay. Other questions?
Hi, Lulu. I want to ask you for your thought about, so normally when we make the disease mutation in mice, sometimes make this heterozygous. So what's your thought about to delete neurexin 2 in particular cell types versus heterozygous mutants of neurexin 2?
So it's homo. It's in humans, heterozygous. Our mice, we only characterize the homo of the deletion because autism deletion, I think it's quite in many different locations, but for particular epilepsy, it's actually quite upstream. So when we delete exon 12, it actually it's a deleterious mutation. So this is where when we do this, we decided to do it this way. Homozygous in the mice, but in human, it's hetero. Hi, Ming.
Hi, Lulu. Great talk. Very exciting. So it looks like neurexin 2 inhibits synapse formation. Do you know how does it do it? What is it inhibiting?
That's great. So how does it do it? Well, I can tell you that we don't know, and that is actually in the more of a neuronal knockout, not just not synapse-specific knockout, because in the synapse-specific knockout in the EMX neurexin 2, we don't really see up to what we so far analyzed, we don't really see a synapse change. But what could it be? I think, of course, when we're looking at synapses, it's not just the pre and post, right? There's also interneurons, there's also different kind of modulator. And more so, I think it's important for us to consider how these are, maybe neurexin 2 is actually aligning the nanocolumn, because neurexin 2, even though they're very similar to neurexin 1 and 3 structurally, but neurexin 2 do have this extra splice site 5. That was my original hypothesis when I was a postdoc. I tested 10 different conditions. However, splice site 5 is actually not the answer. But this extra splice site 5, what's the function? Sometimes I'm not sure. It could be altering the aligning, right? So it could be still at the synapses, but if it does not align the nanocolumn correctly, you might still have all this release based on my data that we see. The release is normal, the postsynaptic is normal. However, why and how we actually get this, it could be inhibiting through, blocking that alignment. I don't know, my guess,
Thank you.
Thank you.
Dong, do you have a question?
You accidentally touched the button.
Okay. If you do, you can unmute and ask the question.
It wasn't just me having technical issue.
Okay. Great. Awesome talks. Thanks, Lulu. Thanks, ChangHui. Great seeing everyone. And
ChangHui, Lulu, great talks.
Thank you so much.
Thank you.
And we'll be back next week. See everyone.
See you all next week.
You.